Hi, my name is Heather Couture, and my pathway map was on Doxorubicin and its role in causing apoptosis in acute myeloid leukemia cells. So, doxorubicin is basically a chemotherapy drug that is used in a variety of cancers, such as breast cancer and acute myeloid leukemia. So it basically, once it enters the cytoplasm through the SLC22A16 transporter, it has multiple pathways or routes it can take to cause apoptosis. One is through the binding of semiquinone, and once it binds to that, it can cause it to become quinone doxorubicin, and it'll go back and forth between doxorubicin that's bound to the semiquinone and then not bound. And through this cycle, reactive oxygen species is produced, and that can go to the mitochondria and nucleus and cause more issues to occur within there with the DNA. Another way it can go is through cleaving the transcription factors, the CREB3L1 transcription factors, which are basically proteins that are bound to the ER or found in the ER, and they play a role in transcription of DNA with the nucleus. But once it cleaves the proteins from the ER, the NH2 group ends up traveling to the nucleus and affecting transcription, just like altering it and just disrupting transcription. A third thing docs can do in the cytoplasm is directly inhibiting topoisomerase 2 in both mitochondria and nucleus. And this is a problem because topoisomerase says one of their main roles is in DNA repair. So if there's like a break in DNA or some kind of just fracture, it can usually fix the code and then reseal it up so that those breaks aren't exposed to things like reactive oxygen species, which would break it up further and cause DNA fragments to be made. And that is bad because DNA is important for the cell, especially division and creating new ones. So by inhibiting the topoisomerase and having things like reactive oxygen species being exposed to it, this can cause torsional stress and DNA breakage, which will lead to cell death, which is what we want in the case of treating patients that have leukemia. An issue is in one of the papers, it discussed how some docs may exit through another transporter called the ABCB1 transporter. And this will basically bring it out of the cytoplasm. And it was indicating that this may be a defense mechanism the cell has in place for this chemotherapy drug to prevent it from causing apoptosis and damaging it further. And it'll go out through that transporter, but it'll go most often will come back in through the first transporter, the LC22A16. So something that I couldn't find in any of the literature was why or like how does doxorubicin get transported back out of the cytoplasm. So, and then why does it get transported out? Which may be a factor of just defense for the cell, but how does it get back out is an issue, because if you're trying to treat a patient with doxorubicin, it's problematic when not all of that drug will stay where it should be and cause the cell death, which is what you want. So figuring out exactly how it gets to the transporter can create methods for scientists and medical professionals to prevent it from going to that transporter. So a guess would be chaperone proteins. So in the case of chaperone proteins are what brings it, we could figure out ways to like deactivate the chaperone proteins or make it unable to bind to the dox. That way it won't go. Or just create other methods just to prevent the dox from leaving the cytoplasm and so it can stay in and just hopefully kill the cell. So that's it.